So without further ado, may I move on to our first speaker this evening, who is one of our excellent award winners. Carol eBay has completed her PhD in plant sciences, has worked for the Cambridge Africa Project, and now the John Innes Centre. And she's going to talk to us about some of the work from her PhD, which involved understanding the nasty things, that's a technical term, for pathogens, which get into rice and can do untold damage, particularly important for food security and particularly in Africa. So Carol, please, if you would like to tell us the story.
Thank you so much. So good evening, everyone. I hope you're all doing okay and safe. It's so great to be here today to present some of the work that I've done in my PhD. I've just finished not too long ago. And it's very exciting to talk about rice and pathogens. So my PhD primarily focused on looking at how rice roots interact with a beneficial fungus, as well as a pathogenic or detrimental fungus, which is called Magnapotha oryzae, a major pathogen that causes rice blast, which is a major disease. And so today, I'm not going to talk about a lot of the work that I've done because I have 10 minutes. I'm just going to talk about just a major highlight.
Carol, could I ask you to move a little nearer the microphone, please?
Okay.
Thank you very much.
Is it better?
That's better, yeah.
Okay. Okay, sorry about that. Okay, so why rice? I chose to study rice because rice is important for global food security. I am originally from Nigeria, and Africa, rice has become a major staple across Africa, unlike how it used to be. There are currently about 1.2 billion people in Africa, and this population is expected to double to 2.4 billion people by 2050. And what this means is that there'll be more mouths to be fed, and a lot of people will be looking for food to eat. Currently, there are about 512 million people living in extreme poverty across Africa, with about 257 million people who are currently hungry. And this is a recent data shared by the SDG Index and Dashboard's report. So like I mentioned, rice is a global food, is a major staple cereal for more than 50% of the world population, but is significantly important for food security in low-income deficit countries, of which many African nations belong to. And so today, I'm not going to talk about the major parts of my PhD, but I really thought I would share more about Magnapotha oryzae, which is called the blast fungus. Magnapotha oryzae is a terror in the rice field. It destroys rice between 30 and even up to 50% of cultivated rice in the field. And so what it does is that is a leaf infection, a leaf pathogen that causes rice blasts through the leaf. However, my PhD focused more on looking at the pathogen and how it gains entry into the plant through the root, which is a non-canonical sort of mode of entry. So rice blast, usually the symptoms will be lesions. It's called necrotic disease lesions on rice leaf, as you will see here. And it can also affect the stem and also the panicle where, the seeds, you see the seeds will not really be produced. And if you look on the right-hand side of the slide, this is a rice field in Louisiana. And so the brown kind of like a seed part of the rice, which is a rice head, are severely infected by this blast fungus. And so they've pretty much lost that field or the production of rice in that field because of this blast fungus. So just very briefly then, my PhD sort of aimed to study the function of rice plasma membrane receptors and how they allow Magnapotha oryzae, which is a rice leaf pathogen, how they allow it entry through the root. And so just briefly, because I know it's a general audience, plasma membrane receptors are also called surface receptors. So they are kind of like receptors that sit on the surface of plant's plasma membrane. So if you look on the right-hand side, this will be the plasma membrane of the plant. And they kind of like sit there as receptors to scan through, looking for microbial signals that they perceive. If they perceive a pathogen or chitin, usually a lot of the fungi have chitin around the cell wall, they will alert the plant and say, hey, induce your defense responses against this pathogen. But if they sense a good signal from a symbiotic fungus, they also will tell the plant, well, get ready, our friend is coming in, open the door. And so they colonize the rice plant. So in this case, the PRRs, or the membrane receptors, would sort of perceive chitin in this case. And I will talk about just CERP1. It's called the chitin elicitor receptor kinase 1. I studied 3 receptor kinases and a receptor-like protein, which is called the CBEP right here, which acts in conjunction with CERP1 to perceive chitin in the case of Magnapotha oryzae, which is a pathogen invading the leaf. Now, in the leaf, what happens is that once CBEP kind of perceives chitin from the cell wall of this pathogen coming in, it would alert CERP1 and form a complex with it. And then they induce defense responses against this pathogen, so they prevent it from coming in as much as they can. But in the root, it is totally different. And it was really an interesting finding, really shocking at first, because I wasn't expecting to see any sort of the phenotype that I saw. I would have expected that CERP1 would act the same way that it would act in the leaf, by sort of protecting the rice plant, but instead it did the opposite. So what I did was looking at this picture right here, this thing that is green, that looks like a worm or a little snake, is actually Magnapotha, is the fungus in the root. And it has been transformed with a green fluorescent protein to allow us to visualize the fungus and keep track of it as it invades the root. And we do this microscopically. And the red is the plant cell wall. It's been stained with a stain called propydium iodide, which allows us to visualize the rice root in the cells and see how the fungus is invading from cell to cell. So what I did was an extensive experiment looking at different time points to see how this fungus is invading in the root in the absence of this receptor-like kinase. So this image that you see here is a wild type plant. So it's a plant that is still in its original form, no modification, and nothing has been knocked out. In other words, CERP1 is intact and the mechanism should be intact in it. And you can see that the fungus moves over from cell to cell, moving happily ever after, 4 days post-inoculation. Now I look at the mutant. So this is a rice plant that we've knocked out CERP1 at 4 days. And when I saw this on the microscope, it was quite intriguing. What I saw here and what you're looking at is actually a fungus that is trapped in a cell. This red part is the part of the hyphae, fungal hyphae, that took up the stain of propydium iodide, which we use to highlight the cell walls. Just to let you know that propydium iodide stains only dead cells, not live cells. So for it to pick up the stain, this fungal hyphae, means that this fungus is actually dead. And if you look up and down, you see that it's trapped. And so I started to wonder, why would this happen? I thought that CERP1 would have sort of alerted the crop that, there's a bad guy coming in, and so induce your defense responses, but instead it did the opposite. And so the way that I could confirm what I saw I did a lot of microscopy to count the number of dead cells I see in the mutant versus the wild type. But also more importantly was to do a gene expression study where I actually quantified the fungal biomass, so the amount of fungus that I have in the root. There's no way you can do that microscopically under the confocal microscope. You can use a marker, molecular marker, and in this case is a Magnapotha specific housekeeping gene that I use to measure. And just real quick, I won't bore you with this. Where you see MOC means that the plant has no fungus in it. The wild type MAG means that this is a wild type plant that has not been, that has everything intact and has the fungus has been inoculated. And the CERP1 is the mutant. MOC means the control, negative control, so it wasn't inoculated. And the MAG is, CERP1 MAG means that the mutant plant has been inoculated. If you look at the graph, you can see that the wild type Magnapotha, you see there is more fungus here if we quantify the constitutively active housekeeping gene, which is actin in this case, compared to CERP1, which in summary tells us that we have a significantly reduced amount of fungus in the mutant compared to the wild type, pointing us towards the fact that CERP1 is actually required to allow or facilitate the invasion of this fungus in rice roots. Now, I did this also at 7 days, following the fungus again from 4 days, looking a little bit beyond, just to see how the plant is responding to this fungus and if there is a catching up in the proliferation of this fungus inside the root. And again, looking at the wild type, you can see that the fungus looks beautiful, looks happy and healthy, invading rice. In fact, the fungus is very clever. It has a way of just not even breaking any cell wall. It just constricts and then gets over from cell to cell. And if you look at the mutant, you can see that the fungus is trapped. It looks really strange and weird. Something had happened there, meaning that there is something going on with the signal, that in the absence of SERC1, the fungus is not thriving inside the root. And just like I did previously, again, molecular quantification just to measure the amount of fungus that we have, because that's the only way we can really decide if there is a reduction in colonization or if there is an increase or if it's the same with the wild type. And what we saw is very similar. Now, again, there is a significant reduction in the amount of fungus in the mutant plant compared to the wild type, which then brought me to the conclusion that SERC1 is required for rice root invasion by Magnapotha oryzae, which is opposite for what SERC1 does in the leaf, where it actually protects the plant against this blast fungus. And the implications this can have for food security is that the information we've provided can help to sort of further research on the development of disease-resistant rice crops, which will ultimately improve global food security. And let me just mention that plant pathogens are very clever. In this case, what we are thinking is that it is possible that Magnapotha, the blast fungus, finds a way to trick the plant into being a friend rather than a foe when it goes through the root. And the plant is not able to discern whether it's a bad guy or a good guy. So it kind of opens up for all the pathogen. It's a biotrophic pathogen. So it has a very clever way of maintaining a biotrophic lifestyle, which means that it appears to be a biotrophic or a mutualistic or endophytic fungus in a way that doesn't come to cause harm. But once it gets into the root, it finds its way through the steel, gets up to the upper aerial parts of the plant, which is above ground, where we think it then causes disease necrotic lesions. And this can be really important for food security, especially in the field. If this is tested in the field, it can help. At the moment, what we think and what is generally known is that rice blast is only caused through leaf infection, and that the spores of this fungus are only dispersed by dew. So in other words, it's not really generally believed that the spores can survive in the soil. But what we see in the lab is different. If this is happening in the field, then it would help us to sort of be on our guard and start to look at the fact that the pathogen may have now adopted a mechanism, maybe from a symbiotic lineage, to trick the plant to gain access into the host cell machinery. So this was part of the PhD. And just real quick, while I was doing a PhD, I was also doing a lot of outreach programs for African researchers and students. I created a program called Reach and Teach Science in Africa. And this is one of the ways that we are applying the research and knowledge, is through knowledge sharing, knowledge dissemination, by providing African researchers who have probably limited resources compared to what we have here, with relevant academic resources that will help them to train others and also to empower the next generation of African researchers. So I'm really thankful to the Department of Plant Sciences, my department, the Cambridge Africa program, and the university in general for supporting this program, which started here in the university in 2017. We usually bring 20 outstanding researchers, PhD students mainly, from different African countries to come to Cambridge. We did it over 2 years, and we decided to go to Africa last year. So by the time I had my CSAR interview, I was actually heading to Benin. And that workshop was very successful. We trained 100 African-based researchers, so mainly early career researchers from about 19 countries in Africa. And here are some of the pictures. We do lectures, we do wet lab training. We also now encourage African researchers to start to design their own lab equipment. And this is a part of the workshop we offer as a labware design workshop. And we do bioinformatics and so many things that we do, professional development as well, to help them on how to write grant applications and all that, just to make them more competitive, and also for them to find a way to utilize what they have to make a difference. And just to in closing is, now because of the COVID-19, we were going to be in Benin again this year in the next 2 weeks. But that is not happening anytime soon. So we're gonna go to Zoom. We're gonna take Zoom to Africa and start to offer online lectures from here, streaming from here in Cambridge, across different disciplines. So if you're interested to offer any course at all, just let me know and we would schedule a time for that to happen. And just in closing, is hunger and poverty is real in Africa. So during the COVID-19 restriction lockdown, a lot of families were starving with children. So I had to just ask a few friends and did some fundraising. We raised almost £4,000 and provided emergency food packages to more than 170 families that are really in need across Uganda and Nigeria. And, it's really dear to my heart. So just to finish and say thank you so much to the CSAR, the organizers. You guys are fantastic. It's been a great opportunity. Also, thank you to my scholarship Gates and the department Cambridge Africa program. I'm currently doing a short-term work for them to design a postdoctoral research fellowship for African scholars. So I'm hoping that in the future, once we get funders to fund it, that we will be able to encourage more African researchers to be able to create independent research, use their research to do, use their knowledge and skills to do research that is relevant to Africa. So thank you so much for your time.
Carol, thank you very much indeed. That was inspiring at so many levels. First of all, that some really deep biological science and plant science can have such a potentially profound effect on the amount of food people have to eat. And then your really exciting outreach work and making sure that the knowledge gets spread around as widely as possible. And I'm sure that's, well, I know that's something the university is extremely keen on. So it's wonderful to have you as an ambassador. I'm not sure if there are any questions. We don't often get many questions at this time, but if there's anything pressing, if not, I'll leap in cheekily and ask one question. The way you describe these fungi, it's almost as if they had a mind of their own. Do we understand the mechanisms that they have for making what appear to be decisions?
Right. Those who study pathogens, like I said, pathogens are very exciting. I mean, they're not good, but they're exciting to study. And that's because those whose specialty are studying plant diseases actually have a way, they've done extensive studies to identify different pathways and mechanisms that the pathogens use to bypass the plant's defense mechanism. One is the production of effectors. So pathogens like Magnapotha in the leaf will produce lots of effectors to sort of counter the plant's defense responses, and that is well studied. But every time there is a mutation, they keep mutating and producing new effectors as the plant continues to try to struggle to be smart to outsmart. So it's kind of like a tug of war. But those who are doing research in that area have done an extensive work and great job to identify most of the effectors. There's still a lot to be done, but so far, we understand that they use effectors and there are other mechanisms that they use to shut down the plant's defense response. And the plant is clever as well. It has a way to also respond back by inducing its own response systems like the receptor kinases to signal and to protect itself in a way.
Fascinating. Well, I shall be much more cautious next time I walk through a field of crops. They've clearly got their eye on people.
Sure.
Thank you again so much. That was
quite
inspiring, as I say.
Thank you so much.
Thank you.